Hello everyone, welcome back to our second topic on ethics in research. In the first topic, I've introduced to you written law and regulation related to bioscience research here in UTM. In the second topic, I'm introducing to you standard practices in related to bioscience research which you should be aware of. Now, unlike the written law and regulation, standard practice is not sourced from any law, but they are practices accepted by the scientific community. Recall this slide from topic 1. We understand now it is important for your research proposal, your experiment design, data collection analysis, and discussion of finding meets the quality that is expected by your scientific field and community. The question now is, what is your scientific field? Who is your scientific community? The peer-reviewed journals that you refer the most is a good indicator that is your scientific community, and that is the source of standard which you should follow. You might ask then, why is it important for you to have a scientific standard and a scientific community? Now, research progress only when the findings are verified by others. This is the first reason why researchers publish their work. The same goes with you too. Your work, your research will only progress if other researchers in your scientific community are able to replicate it. In other words, reproducibility is validity. Findings that are reproducible means they are nearest to the facts. Vice versa, findings that are not reproducible means there are no evidence they are actually close to the facts. Why are researchers worried about irreproducible findings? It is because it impedes scientific progress and is a waste of time and money. Importantly, it can erode the public trust in science. Now that's mean the first quality that you must have in your work is replicability and reproducibility. Replicability is obtaining consistent results using the same input of data, the same methods and condition of analysis, and this is done by the same lab, the same setting, and the same people, which is you, or people in your lab. On the other hand, reproducibility is obtaining consistent results using the same input of data, the same methods and condition of analysis, only now they are carried out by different people in a different lab in a different setting. So what are the standards that you must follow in order that your work has this quality? One of it to write your methods in a detailed and correct manner so that the other people can replicate your work. This is an example of methods that you should follow if your work is related to getting samples from the environment. There must be adequate description of the site where the sample was taken and the explanation of the sample itself and how the samples are kept and where they are kept. It must also have adequate information on what are the reagents used and where they are from. Same goes with the equipment that are used, what are they are being used, and who are they made from. Now, the second quality that your work must have is objective discussion or unbiased discussion. Now, objective discussion means is committing to facts, whether this is in favor of or against your own hypothesis and belief. In other words, objective discussion commits to the facts of the finding itself. Now, it must be free from your own interest or your own personal value or from others people's interests. A good indicator that suggests a discussion is not objective or biased is a cherry pick of what result to discuss and what result to hide. So what is the consequence of biased discussion or unobjective discussion? We will stop this topic to here for a homework. Now watch this documentary and then reflect back on the project for your research in your postgraduate study. Now discuss what is the impact of an objective discussion or biased discussion of your research project on your publication and the postgraduate student that is going to continue your work. Now submit your discussion in the e-learning link. And this is where we end topic 2.